The next speaker is Ruud Delwel from Erasmus Medical Center in Rotterdam, who will talk about EVI1 and leukemia.
Well, thank you very much, Hugues, for giving me the chance to discuss with you some of our data. So we have become very interested, not so much in promoters, but in enhancers. And my talk will be about enhancers and the role of enhancers in leukemia. And it all started with, does not work? Okay, to do this. So, acute myeloid leukemia, trying to understand the molecular biology of acute myeloid leukemia. So, acute myeloid leukemia is not one disease. AML is subdivided in many, many different subclasses. And every subclass has its own molecular defects and background. But it's important that these recurrent genetic and cytogenetic abnormalities, they are predictive for therapy outcome, and they are being used throughout the world as major decisions whether patients get transplantation, yes or no, after chemotherapy. Biologically, we know that many of the cytogenetic abnormalities usually are characterized by the generation of fusion genes, right? Recurrent translocation give fusion genes. Today, however, I'm going to talk first of all about a type of leukemia with a recurrent abnormality, which is the abnormality here indicated here, 3q26, 3q21. Recognized by WHO as a refractory type of leukemia. These leukemias are completely insensitive to chemotherapy, and as you can see here, these leukemias really don't respond, and every patient that has this abnormality dies of the disease. There's just no cure for them. We have overexpression of EVI1, the EVI1. It's located at 3q26 and was first identified as a retroviral integration site. EVI1 stands for ectotopic viral integration number 1, so in mouse leukemias. At the 3q21 site, we always thought there was the RPN1 riboforin 1 gene, and these leukemias are also called RPN1 EVI1 up till 1 or 2 years ago, and now the name has been changed, as you will find out today. There are two flavors of 3q21, 3q26. There are either inversions or translocations. They always involve the same regions, but we had no clue really where the regions exactly were and the translocations were. EVI1 encodes for a zinc finger protein. That's the only biology I give you about EVI1. It's a zinc finger protein and expressed in the nucleus, and most likely acts as a repressor of transcription. And finally, what is important, when you knock EVI1 down in cell lines that express it, the cells die. What you see here, these are two EVI1 positive leukemias. This is an old-fashioned shRNA experiment. What you can see, you get complete loss of colony growth, and the cells differentiate really frequently, and they die. So this was the hypothesis when we started. The idea was that there is an 3q21, there is RPN1, here's EVI1. EVI1 is expressed in very, very primitive hematopoietic stem cells, which are not in cycle. The moment they cycle, they lose EVI1. But in those leukemias now, upon translocation, EVI1 is upregulated, and we predicted there might be some regulatory element activating EVI1 expression, because we knew there was no fusion gene, as in contrast to many of the other translocations. So we decided to really find out what is going on there. And first thing that we had to do is, where are the exact breakpoints at the 3q21 site and at the 3q26 site? Where are the breakpoints near EVI1, and where are the breakpoints near RPN1, and what is the minimal region that always translocates to EVI1? So how did we do that? We took 41 cases with this abnormality, isolated DNA, did capture sequencing, taking the 3q21 region, 3q26 region, deep sequencing, and then found out, where the breakpoints were. So this is what we found. Here we have EVI1, MDS1, not important for today, but it is an important gene as well, but not for today. Another gene, and here you find all the inversion breakpoints, and here all the translocation breakpoints. All translocation breakpoints were 5' of the gene, all the inversions were 3' of the gene. If you would draw what's going on, you would understand why this is. But it already pointed us that, first of all, definitely there's no fusion gene, but the fusion gene would also always expect it would be on either 5' or 3'. And it already points to the possibility that an enhancer is translocating, because enhancers act either 3' or 5' of promoters of genes. So what happened on the other side? On the other side, we didn't find this very nice distribution between translocation inversion, but we saw that all the breakpoints were between the GATA2 and RPN1 gene, and this is approximately 125,000 base pairs. Okay? So the question now is, what is the minimal region that always translocates from here to EVR1? Okay? So draw it in a little different way. So here you see now really, this is EVR1, no, RPN1, and here there's the breakpoint, and here at the breakpoint now you start reading EVR1. That's really what is, sorry, this is what's happening. And you see this in all those cases. So really the smallest region that always translocates in every patient with this abnormality is this region of 18 kb between the RPN1 gene and the closest breakpoint in this particular patient. So we next wondered, what's going on here. So we asked the question, is this region that now is translocating, is this active? Can we identify enhancers there? So one of the things, simple things to do is HCK27 acetylation pull-downs, which is a mark, as we already heard today, of active chromatin. So we did this, and what you can see here, so we did HCK27 acetylation pull-down and then deep sequencing, and what you see here, this is 27, just only look at the blue one here. You see that this region is highly active, highly open chromatin, 27 acetylation. Acetylation occurs through enzymes, and one of those enzymes is P300, which is really a mark for enhancer. So we did P300 chip sequencing as well. And then we found in this region 2 large P300 peaks, okay? Really suggesting that one of these regions are really the enhancers translocated and activate EVR1 expression. Another way to show whether there is true involvement of that region with the promoter is so-called 4C sequencing. So the whole idea is that you get looping of the enhancer to the promoter, and there's a certain way which is 4C sequencing by which you can analyze whether there are particular DNA regions that interact with the promoter of interest. And the way you do it is this. So you take your cells, you cross-link the DNA, you digest DNA, and regions that are very close to your promoter are now linked to this region. Next thing you do, you religate the DNA and then design primers, which you call the viewpoint, from which you now start analyzing whether I find regions that have been formed, in this artificial circle that may have been interacting with my region. And so you do then again, deep sequencing. And when we did this, we found this here. So this is the viewpoint. Here you see very nice, a peak, which is the EVR1 peak, okay? And there's another small peak here very close to the RPN1 locus, where really what happened, of course, is there was an inversion, so this thing was really here very close to the EVR1 gene. But there was a clear dip here, and so we got 2 peaks, so a region that really forms a complex with the EVR1 promoter. So we did this in 7 independent cases and gave the data to bioinformaticians, and they calculated that right on top of this P300 peak, there was the highest interaction with the EVR1 promoter the moment you get this translocation. So the next question, of course, is this enhancer truly involved? So this experiment already was 4 or 5 years ago. This was just, first we did a TALENS experiment, which was very popular for only half a year, until then CRISPR showed up. And we did both TALENS and CRISPR experiments. To make a long story short, this was a TALENS experiment where we really generated many, many clones in a cell line with a 3q6 abnormality, trying to digest this one P300 peak out. And in fact, we got 4 clones in this experiment. And as you can see, really, so we delete this one P300 region. Now, really, in the translocation, this region is still 250 kb away from the promoter of EVR1, really far away, okay? You delete it in the 4 clones. You see this is control clones. These are clones that were treated the same, but not successfully. You see very nice EVR1 expression, and you completely lose the EVR1 expression in those leukemias. Whereas GATA2, RPN1 on the other side, nothing happens to it. I already told you, EVR1 is very important. If you knock it down, the cells stop growing. So what happened here, and we see also here that the cells, we could harvest a few cells that we used for this RNA sequencing. But what you can see very nicely is that the cells really stop growing, and I can tell you that the cells very nicely differentiate because this one cell line, it's MUTS. It's called MUTS, which is a 3q6, 3q1 cell line. It's CD34 positive, but it does differentiate a bit, as you can see here. But the moment you cut out the enhancer, or you knock down EVR1, you see that they lose CD34 and they start differentiating and they die. So We still had the idea that this was an enhancer of RPN1, but then really want to know, what the enhancer really does. And so we did a very simple CRISPR experiment this time using K562. If you want to do CRISPR experiments and you want a cell line, which always works, use K562. Well, the good thing was, this cell line does express the two genes that are located here, RPN1 and GATA2. So we cut this P300 site out in K562 and then asked the question, what happens to RPN1? Nothing. But this is what happened. It was a long-distance enhancer of GATA2. And so really, what we have now, and I finished this part here, is that in those leukemias, so there's a normal allele and an abnormal allele. The normal allele and in normal cells, usually in bone marrow cells, there's GATA2 expression driven by this enhancer. There's no EVI1, but upon translocation, EVI1 is expressed, and we have shown very nicely that every leukemia with this abnormality has only one GATA2 allele on, and it loses its GATA2 allele. So the conclusion was here is that we have a breakpoint in these leukemias. These are unique and affect a crucial non-coding regulatory 3q21 region. So we show that one chromosomal rearrangement causes two events, which is activation of EVI1 and repression of GATA2. And there is quite some evidence that GATA2 loss is really important. There are families with one allele GATA2, and these families really very frequently develop acute myeloid leukemia with monosomy 7. Whether they have 3q, we don't know. One recent experiment, studied by, published by Masashi Yamamoto and his team, who in fact found exactly the same thing as we found, but with a completely different model. He showed that when he crossed a 3q21, 3q6 mouse with monolithic GATA2 mice, these mice develop leukemia much more quicker and frequently than mice that only have 3q6 EVI1, really suggesting that there is really a clear synergy between monolithic GATA2 and overexpression of EVI1. So this is the situation now. We think we have a unique situation here, which is an EVI1 promoter forming a complex with the GATA2 enhancer and a unique, hopefully a unique set of factors. Common factors which are also bound probably between GATA2 promoter and enhancer, but hopefully also unique ones. And so we have three questions that we try to address now. So first of all, is the transcriptional control of EVI1 sensitive to normal regulation? Second question, what is the mechanism of EVI1 transcription, and can we uncover unique targets to deregulate EVI1 expression? So what I show you now is, more lab meeting stuff, because we are still on the way with many things. So one thing that we did now is ask the question, is EVI1, can it still be regulated? So I showed you already this cell line, it's called MUD cell line. It has 1, 2, 3 fractions here, and we just sorted those fractions. And ask the question, is EVI1 expressed here, but is it also still expressed here, or does it go down? And so we have a very nice immunostaining with an antibody that you can use for EVI1. And what you can see here in the unsorted fraction, very nice EVI1 expression. If we take the CD34 plus, CD15 minus in the most immature cells, we still have EVI1 expression, but when the cells mature, they lose EVI1. So, and we did FISH, even it's a cell line, we did FISH, and definitely the translocation is there. And we also showed that, in several primary AMLs already now, that even though they have 3q6 abnormality, there's EVI1 expression, EVI1 can be turned down. And that's going to be shown here. And so then the other question, of course, is it cause or consequence? And we think it's cause. So if you really knock down EVI1, I already showed you this experiment, but whatever we do to knock down EVI1, we see this thing happening, right? So first EVI1 goes down, and then we see very nice induction of differentiation. So we think it might be very important to really find out how EVI1 is regulated, and can we maybe target this thing, because otherwise we have nothing, in fact, to cure those patients. So other thing that we did, so we looked through, back in literature and in our data that we have an enormous patient bank, and also in collaboration with others. There are very rare cases of, for instance, translocation 2-3, 3-6, 3-7, all with EVI1 expression, but now not with GATA2 as the enhancer driving the expression. So we asked the question, is there a common mechanism of EVI1 transactivation in those leukemias? And as I said, this is just data that's underway, but what we did again those captures. So we got more than 100 AMLs with all these different translocations. we did capture of the 3Q region, and then and then sequenced, and hopefully, got at a breakpoint, and then sequenced into the partner region. So, just give you one example. These are all the cases with 3.8. You see that very nice breakpoints are all 5' of EVI1. And what we found in every patient with a 3.8, we get into the MYC locus. And really what we found is that here is this so-called MYC super enhancer that many people are working on nowadays. And in fact, we have even one breakpoint right in the middle here, and we could even predict which enhancer of those 5 small enhancers that are there is most likely activating EVI1 expression. So we did this for all those cases, and we found for all of them the putative target gene. And if you really look carefully to them, these are all genes that are expressed in very primitive hemopoietic progenitors, and many of them are also transcription factors. These 3 here are really probably a completely different mechanism. Here we do have a fusion gene, okay? This is EVI1 NRIP, EVI1 ETV6, and EVI1 RUNX. In fact, the other way around. But anyway, these are fusion genes. These are not, and we predict that and we could define in all of them a potential enhancer that always translocates to EVI1. So what we really would like to do now is, this is the hypothesis. EVI1 translocates to genes normally expressed in hemopoietic stem cells. Enhancer-containing regions translocate to EVI1, and these translocated enhancers bind to the heptad. That's what we found. So all the primitive hemopoietic transcription factors bind to all the enhancers that we identified. So the translocated enhancers appear to be ACK27 hyperactive. I haven't talked about this yet. The GATA2 enhancer that we showed translocates to EVI1, turns into a so-called super enhancer. It's a typical enhancer, which is a small region of 27 acetylation, but when you get a breakpoint, it's a huge region of sometimes 40, 50, or maybe larger kbs of 27 acetylation. In every case that we look now, we find that a sort of a super enhancer activates EVI1. So the hypothesis is that a common oncogenic mechanism of enhancer-driven overexpression is occurring in these patients with EVI1, and we really would like to find out, so what are the protein binding motifs in all these enhancers? What do they have in common? What are the transcription factor complexes, and what are the epigenetic regulators? So to do that, we are currently developing a model, and we have developed a model, and we now have the model working, which we would like to use for screening purposes. So what we did, we went back to our cell line and cloned 3' of EVI1 and EGFP, and with a P2A cleavage site. So what this means is that you have one transcript, but 2 proteins coming from it. So because you never know what the protein is going to do if you have a fusion EVI1 EGFP. So that's what we did. So we have only EVI1 and EGFP, but now we have a very nice EGFP positive line, and we could really show that by all the things that you have to do, that really GFP is really in line here with EVI1. And if you do, for instance, an experiment where you do an EVI1 knockdown, upon EVI1 knockdown, you see very nice GFP going down. So really we can use this GFP staining as a measure of turning on or off EVI1. So does it work if we do the enhancer deletion? So now we now also have an inducible Cas9 in that line. And so here you see that here you have GFP expression if you do nothing, if you give docs, if you give only guides, if you give docs plus guides, cut out the enhancer, you see very nice GFP going down again. So we can really do a nice guide screen, to really identify regions in our enhancer that are really essential for activating EVI1 expression. And here we do it also for anti-EVI1 to really show that this GFP loss goes hand in hand with loss of EVI1 staining. So does it really work? So then what we did, we sorted, upon knocking down this enhancer, we sorted the GFP negative, the intermediate, and the high fraction, and then did an EVI1 qPCR, very simple experiment, and see very nicely that the EVI1 negatives are here and the EVI1 positives are still in the positive side here. And if you do the PCR to really show that you did cut out the enhancer, you see very nicely, here we cut out the enhancer, here the positive ones were not hit, and the enhancer is still there, and we couldn't cut it out. So this model now will be used to carry out motif disruptions in this translocated enhancer by genome editing. We will, of course, also use all the enhancers that we now identified and ask the question, what do we have in common? And common motives will be targeted, but we will also do a scanning experiment using the whole super enhancer and really try to find out what the essential regions are. We would like to uncover regulators EVI1 transcription by doing shRNA and most likely, really go for guide RNA Cas9 screen to identify target proteins that activate EVI1 expression, or identify inhibitors by doing a compound screen. And with that, I think, am I?
You still have some time if you want.
Yeah, sure?
Yeah, absolutely.
Okay. Then I go to another enhancer story. Try to do this quickly, okay? So this was my EVI1 part. There's another gene that we have gone very interested in, also because of enhancers. It's a gene called CBP-alpha. CBP-alpha is frequently mutated in acute myeloid leukemia, and it's a gene that is absolutely essential for hematopoietic development. CBP-alpha is essential for the development of many tissues, okay, which is important for my talk. And in hematopoiesis, it's turned on in hematopoietic stem cells, and the moment it turns on, cells are driven to become myeloid, become neutrophils. That's really what is CBP-alpha doing. Knockouts have been made, and you have only blasts. You get no neutrophils development anymore. And as I said, it's frequently involved in leukemia development as well, in many ways. The question is, what we didn't know, is there any evidence of aberrant transcriptional control of CBP-alpha in leukemia? So if that is the case, then we have to define myeloid-specific enhancers in CBP-alpha. So we did again 27 acetylation pull-down. And what you see here, this is the CBP-alpha locus. This is an SLC, which is a household gene, CBP-alpha, CBP-gamma. This is a region of approximately 175 kb. This 27 acetylation overlay, you see many peaks here. And every peak, I can tell you, is a potential enhancer for CBP-alpha. How do we know? This overlay comes from this total experiment. This is 27 acetylation analysis of this region in all tissues that express CBP-alpha, and these are the tissues that do not express CBP-alpha. Here you see the region is completely closed. There's no 27 acetylation anywhere. Here you see there are many peaks, and what you can see very nicely is that every tissue has its own combination of regions turned on. And to make a long story short, monocytes and neutrophils, look here, there are two peaks here, plus 42, plus 34, that they're only present here, and you don't find them in any other tissue, although these tissues do express CBP-alpha, okay? So we predicted that these enhancers are essential for myeloid development. If you look at CD34 positive cells, the primitive cells, you see only two. There's the +9, which is an enhancer which is ubiquitously active in every tissue, and the +42 is really the one that is still active also in primitive, is the first one to be turned on there. This one contains many motifs for very primitive hematopoietic transcription factors. And if you look at data from John Pimanda, pull-downs in chip sequencing in CD34 positive cells for GATA2, FLY1, RUNX, etc., you see that there is the binding. It's not at the promoter, nowhere else. Only at this +42 you find binding of all these hematopoietic transcription factors. P300, also major binding here. How important is this? So in mice, there is a very identical, it's almost 90% identical enhancer, which is not the +42, but it's a +37. So what we generated knockouts of this enhancer. Well, what I didn't tell you, the straight knockout has a problem. You have no myeloid cells, but the mice die within 48 hours after birth because of long liver failure, okay? These mice were happy and alive. No enhancer, but CBP-alpha really dramatically goes down in the bone marrow, whereas in other tissues, nothing happens with CBP-alpha, okay? And then when you look at neutrophils, you see very nicely here and here, wild type, complete loss of neutrophils, which are the MAC1, TR1 double positive cells. So, really seems that if you target this enhancer, you have a leukemia type of phenotype, you get blast cells, the cells cannot differentiate anymore. So we argued, maybe there is something going on in some patients with AML. When we look at this, let's have a look at the enhancer. So we took almost now 2,000 AML patients, we did qPCR, and so we have here 10% of leukemias with very low expression of CBP-alpha. So these are the phenotypes of those leukemias. I'll come back to that in a minute. So we did capture sequencing of this whole region of all the low expressors, hoping to find patients, with abnormalities in enhancers. We never found one single one of them. We had one patient that has a complete loss of the entire TAT. The entire CBP alpha locus was completely gone on both alleles. But that was it. But what we found here, and that's the last group I would like to talk about, is a group of patients which has translocation A21. It has the AML1 ETO gene by which it has been transformed. So this is a large gene expression profiling study that we did years ago. And what you can see here on this profiling, these are all the patients with an AML1 ETO, translocation A21, and you can see they have very low CBP alpha expression. In fact, if you look at it in a different way, these are the low expressors here. So we asked the question, does AML1 ETO cause downregulation of CBP alpha? So this is an inducible model where you activate AML1 ETO in a U937 cell. The moment you do that, you see that CBP alpha goes down. There really appears to be a relation between AML1 ETO expression and CBP alpha going down. When we introduce it in CD34 positive cord blood, and you compare it, for instance, with MLL-AF9, you see very nicely CBP alpha goes down. So the question is, does AML1 ETO interact with any regulatory element in that locus? So AML1 ETO has a RUNX binding domain, and then there's another domain here, come back to that later. Did a pull down here in a cell line, primary AML cells, and again, what do we see? AML1 ETO binds to the plus 42 KB enhancer that we identified, that if you delete it, then you have no CBP alpha expression anymore. There are 4 RUNX binding sites, really most likely causing the AML1 ETO to bind there. And what we do know about ETO is that the ETO moiety really recruits HDACs, which are the enzymes that deacetylate, in fact, regions where it is, that are in close proximity. So this was the hypothesis. Normally you get activation. Now these are replaced by AML1 ETO, you get HDACs, and with that you get deacetylation of the region. So is this really happening? So look what we see here. These are again the CD34 positive cells where we either introduce MLL-AF9 or AML1 ETO. You see very nice deacetylation of this region. And again, here this is where AML1 ETO binds. And remember, these are the cases with low CBP alpha expression. And if you take a control region, it's not a technical thing because in other regions we still have very nice acetylation. Okay. So then the question is, does knockdown of AML1 ETO reverse acetylation of the CBP alpha locus? And we did this experiment together with Connie Bonifair, plus 42 KB. Here you see when you knock down AML1 ETO, you don't have any binding of AML1 ETO anymore. And if you then look at ACK27, in this case K9 acetylation, you very nicely gain K9 acetylation back at this locus. We did the same for the 27 acetylation region, and you see very nicely, this is the control. Upon AML1 ETO knockdown, you gain acetylation back of that region. And what about primary AML samples? We looked in primary AML samples. So these are control AML samples with other abnormalities. These are AML1 ETO cases with very low CBP alpha expression. And you can very nicely see there's much less acetylation of this entire region here. So finally, we asked the question, does knockdown of AML1 ETO reverse the enhancer CBP alpha interaction? Remember, promoter and enhancer might interact with each other. So again, here we did a 4C experiment, and now asked the question, is there a difference in the interaction between the enhancer and the promoter of CBP alpha? So we took 3 patients with AML1 ETO and 4 patients which are with another abnormality. This is the viewpoint, this is the plus 42 enhancer, and what you can see here, there are 2 peaks here. In blue is the AML1 ETO, and these are the controls. This region forms a contact with the plus 42 enhancer, but this region loses the interaction in those patients. And we showed here a little bit better. So the whole idea is that in AML1 ETO, this interaction is lost, whereas this is present in normal hemopoietic progenitors or in other AMLs without an AML1 ETO abnormality. So this is my final slide. What we think is going on in normal situation, there's a plus 42 KB enhancer, which is essential for neutrophil development. There's interaction between this and the CBP alpha promoter. You get acetylation, CBP alpha goes up, and you get neutrophils. But in certain AMLs with AML1 ETO, the interaction is lost. You lose your 27 and K9 acetylation, CBP alpha goes down, and you have no neutrophils anymore. You have a block of differentiation. So with that, I hope I've convinced you that enhancer biology is exciting. At least that's what we find. And these are all the people that are involved in our work, many, and particularly this guy, Roberto Avellino and Stefan Greuschel, were the people that were really majorly involved in the work that I presented today. Thank you very much.